I'm really sorry about that, guys. Technical difficulties. So this is going to be part 2 of section 17.1. Sorry about that. So we were talking about plasmids or cloning vectors and what they have, and now we are specifically using bacterial plasmids we're just analyzing them a little bit more, and we're using them as our main example. So any kind of bacterial plasmid itself, right, it's an extrachromosomal circular DNA. It has its own origin of replication, its own little ori, and so that's why it can replicate independently of what the actual bacterial chromosome does, right? In addition, we have specifically engineered these bacterial plasmids to gain things that we want them to have. For example, we put in, I'm going to use the same color. We put in a multi-cloning site, which is basically full of restriction enzyme recognition sites or restriction sites. Okay, so many different restriction sites. So some for like restriction enzyme EcoR1, some for restriction enzymes ALU1, etc., etc., etc. We have all sorts of a variety of restriction sites that we can choose to cut, right, and to open up our vector so it becomes a C, and then basically put in our new target DNA. And then this restriction enzyme site is usually found wedged in between an actual functioning gene. It's a functioning gene that acts a little bit like a selectable marker, okay? But this selectable marker gene is necessary for us to see if the plasmid has actually obtained the DNA fragment or not. So again, as I proceed now with the explanation, that should make more sense. So this right here, okay, this right here is a selectable marker gene. One of the ones that we use the most is called the LAC-Z gene. You guys are already familiar with the LAC-Z gene. This encodes beta-galactosidase. All right, and then in between, basically wedged within the LAC-Z gene, the functional LAC-Z gene, this is a completely functional LAC-Z gene, we have the multi-cloning site right here, which has a bunch of restriction sites or recognition sites. In addition, we have another gene that acts again as a selectable marker, but this selectable marker is basically to see whether the organism we put the bacterial plasmid in, which is usually another bacteria, usually E. coli, since it's the most easily manipulated in the lab. We want to see if the E. coli actually took up the plasmid in the first place, and so we need to give the E. coli a gene that it normally doesn't have. So this would be ampicillin, like this is an example, ampicillin resistance gene. So normally E. coli is not resistant to ampicillin. So if we grow it in a medium with ampicillin, then the bacteria is going to die. But if this plasmid successfully goes inside of the bacteria, when it goes inside the bacteria, it takes all of its genes with it, right? And so it's going to take the ampicillin resistance gene as well, express it. And so now this bacterium that has taken up this plasmid actually is able to express the ampicillin resistance gene and therefore become resistant to ampicillin. So in a media that actually contains ampicillin, this particular bacteria with the plasmid can actually resist it. So it'll be explained in a little bit more detail as we continue, okay? So let's pretend now we have a particular target DNA that we want to put in. Let's say it's human DNA, because why not, right? So it's human DNA. Human DNA, you guys know our genome is absolutely huge, disgustingly huge, right? But we only want a specific target DNA, this particular gene. Let's imagine this to be the insulin gene, since it's something that's done very often, okay? The insulin gene is right here, and this is the rest of the DNA, which I'm not even gonna bother to draw out because there's just so much of it, right? So how do we specifically isolate this target DNA? We cut it, we basically find restriction sites at that flank the target DNA, we cut it, right? We cut it and basically take that sole target DNA out. We do this basically through a process also known as like gel electrophoresis. There's a variety of different ways we can specifically isolate one target gene from the rest of the genome, right? We also use things like PCR to do so. That will come in the following sections. So here we have our target DNA. This is the DNA that we want to put in. Let's pretend we cut with a very common enzyme. right? Restriction enzyme, EcoR1. So we saw that EcoR1 has sticky ends. I don't actually remember what those sticky ends are, like the actual sequence. So if I don't remember them, I don't expect you guys to remember the sequence of the restriction enzymes. That's completely berserk. You're always going to be able to see, and like if you're ever in the laboratory and you have these restriction enzymes at disposal, it will be shown on a little tube what the sequence of the restriction enzyme actually is, how it cuts and what sequence, the recognition site it cuts. So we cut our target DNA like so, and we've completely taken it out. Now the next step is we want to put it into our cloning vector. So we go ahead and we cut open our cloning vector. And if you see here, when our cloning vector gets cut open, it gets cut open in the multi-cloning site, which is found within the LAC-Z gene, right? So our LAC-Z gene at the moment is broken up. We have part of it over here and the other part over here. But we've also cut this with EcoR1, right? So we've given it the same sticky ends, complementary ends as the ones that are complementary to the ends of our target DNA. We've also cut this particular enzyme with EcoR1, and we now obtain this. Okay, all the other things, all the other features stay there. They don't go anywhere, right? We have our ampicillin-resistant gene, and we have our ORI. Everything is the same. We've just cut open one of the restriction sites in that multi-cloning site. All right, so now we put the target DNA and the plasmid in the same tube. Okay, and we hope that these two ends will anneal. So this end will anneal to this end, and this end will anneal to this end right here. And we also add DNA ligase to basically secure, to form that covalent bond and the phosphodiester bond between the nucleotide sugar phosphate backbone. Okay, so two possible things can happen. One of them can be successful. So if I now add, if I do plus, nope, like that, there we go. If I now add my target DNA in the little tube and the DNA ligase that we need and the proper conditions and temperatures that we need, two things can happen. One of them can be a successful outcome, okay, where our target DNA goes inside and actually becomes part of the cloning vector. So it would look something like so. That is a terrible looking vector. Sorry, guys. I'm trying. Okay, and so this sticky end of this target DNA would be complementary to this sticky end. Here's my target DNA, and then the opposite sticky end being complementary there. So it's a very terrible drawing, I'm sorry. But essentially here I have my target DNA, right, inside my cloning vector. And so my cloning vector has the ampicillin-resistant gene still, the ORI is still there, and now that LAC-Z gene, which was previously intact, but now since we put something in the middle of the LAC-Z gene, our LAC-Z gene is interrupted. So, as you can imagine, our LAC-Z gene does not work. It is now non-functional. It cannot be expressed properly to make beta-galactosidase. So this particular plasmid with our target DNA inside of it doesn't make beta-galactosidase. And this is really important because this helps us screen the plasmids that have taken up the target DNA against the plasmids that have not taken up DNA, because that is our possible second scenario, right? And the second thing that can happen is that during annealing, instead of this sequence right here coming together with this sequence and this one coming together with that, why doesn't sometimes, this happens randomly, the plasmid DNA can just kind of link up, because these two sequences are technically still complementary to each other. So the plasmid DNA can just join again, link up, and basically go through a process known as self-ligation. It just ligates back to itself. And so we have the other possible scenarios that we have a plasmid that is fully intact, that has not taken up our DNA, and that has a completely intact LAC-Z gene. And therefore, these guys do have the beta-galactosidase gene. See how it's different, right? Okay, they don't have the target DNA inside of them, so they do have the presence of the beta-galactosidase gene. So this is really important because when we grow them in a media, when we actually allow the bacteria or the host organism to uptake the plasmids, when we grow them in the medium, we can differentiate between which bacteria has the plasmid with the DNA and which bacteria has a plasmid without the DNA. depending on the presence or absence of this particular enzyme, beta-galactosidase. Okay, so now that we have this, in our tube, we have both plasmids that don't have the target DNA and plasmids that do have the target DNA. At this point, we cannot differentiate between them just yet. We now have to, I'm going to draw on another piece of paper. We now have to go through a process known as transformation, and you actually have a lab called the transformation lab. This is what you're doing. You're putting a vector, a plasmid vector, inside a bacteria. So it's inside any kind of host cell. This host cell is normally a bacteria. It could also be yeast. Yeast, there we go. Saccharomyces cerevisiae is a very common yeast that we use, and it's kind of, it gives us another ulterior, non-prokaryotic alternation. Nope, that's not the alternative. That's the word I'm looking for. So it gives us that eukaryotic alternative, which is very important sometimes. Okay, because eukaryotes process things a little bit differently. So transformation can happen in two ways. We can either add like calcium ions to our bacteria and then go through a process known as heat shock, where we literally put the bacteria in a really hot environment, in a hot water bath for a few seconds, and then take it off and then put it directly in the ice. So completely shocking the heat, shocking the bacteria. When that happens, the plasma membrane of the bacteria, which is normally nice and intact. So here's my bacteria with its, I'm actually going to draw the bacteria in orange, right? This is a bacteria, and this is the normal genome of the bacteria, nice and circular, as we know most bacterial chromosomes are. Okay, when this bacteria is exposed to calcium and then heat shock, calcium treatment and then heat shock, the plasma membrane gets a little bit wobbly, gets a little bit loosey-goosey. So you kind of start having like pores start forming in the plasma membrane, okay? Pores start forming in the plasma membrane. It becomes quite permeable to the things that are outside. So as soon as our bacteria is in this state, we add the tube with the plasmid. So here's my tube. Whoa, that's my tube. Okay, some tubes have just the so inside the tube, some plasmids are just plasmid, and then other plasmids have the target DNA in it. Okay, so hopefully you saw that. So we add this inside towards the bacteria, and we hope the bacteria takes up some of these plasmids. So what are the possible three scenarios in this case? The three scenarios in this case are, oh, hold on, sorry. And then the other type of transformation that we can have is called electroporation, which essentially achieves the same thing, except through different little pulses of electric, like voltage, kind of like pulsating the bacteria, and then the bacteria will form like this permeable plasma membrane so that these plasmids can go inside. So it's called electroporation. This is all in your slides, guys. It's easier for me to draw and then read off the slides. I prefer that. Okay, so once transformation occurs, this also has to happen to bacterial cells that are competent. So competent cells, I believe your slides go over this, but competent cells are cells that are actually able to take up outside DNA, right? Most cells are not competent. Most bacterial cells are not competent, though there are some that are naturally competent, and these are the bacteria that are terrifying to me. Like Streptococcus, for example, is naturally competent. It can just take up DNA from the random environment. Meaning, I'm going to try and switch it up a little bit and change my camera with you. So it could take up, if a bacteria has died, all of the insides of the bacteria are now in the environment. And if we have a competent bacteria in that same environment, that competent bacteria is capable of picking up the DNA that the previous bacteria just let go of because it's dead. So these kind of competent natural bacteria are terrifying because they can just take up any kind of random DNA, like naked DNA from the outside world, right? And we know that's called transformation. Okay, right? So some cells are bacterial cells are naturally competent. Other cells, like E. coli, for example, are not naturally competent. So we have to make them competent. And this is basically, this transformation pathway is how we do it, right? This method of transformation. Let's see how I talk about this in the slides, or if I do. I do not. I don't think, because it would have been something that would have been in bold. It's important though, because if the cell is incompetent, it's not going to take anything up. Okay. Anyways, so as I was saying, let's go back to my. Let me think. Okay, so now where's my? Yeah, there we go. So now the three possible scenarios that can come when we transform the competent bacteria cell with plasmids, three things can occur, okay? Three things. Some bacteria may not take up any plasmid. That happens. Once the conditions are returned to normal, the plasma membrane patches itself back up, and randomly enough, no plasmids are obtained. That can happen. Other bacteria do take up the plasmid, but maybe they take up just the plasmid that doesn't have any DNA fragment in it. That also can happen. And then the third possible scenario is that our bacteria takes up the plasmid, and amazingly enough, it takes up the actual plasmid with the DNA fragment in it. Okay, hopefully you can see the difference in color there. All right, so then the next step is, now that we have all these bacteria in the same tube, and we can't differentiate the bacteria in simply a tube of broth, we need to plate them on a specific media. So we plate all of these bacteria, since we don't know which one's which, on a media that has ampicillin. It has ampicillin, and it also has another substrate known as X-gal. X-gal stands for a really long name. It's like, I actually don't even know. It's like 5-bromochloroglucuronidine, something crazy. Basically, X-gal is an analog of lactose. And we know that beta-galactosidase works on lactose. X-gal, when beta-galactosidase is present, okay, X-gal, which is usually a colorless substrate, becomes blue. Okay, when beta-galactosidase is not present, X-gal, which is colorless, it doesn't get worked on, so it remains colorless, and we still get that white colorless color, pretty much. Okay, so X-gal and ampicillin media. So our bacteria are growing on it. Only the bacteria with the plasmid are going to be able to grow in an ampicillin-ridden or an ampicillin media, because this plasmids, as you remember, have the ampicillin-resistant gene. I didn't color it in here, but they do both have the ampicillin-resistant gene. Nowhere on the bacterial chromosome do we have an ampicillin-resistant gene. So these kind of bacteria, as soon as we put them in the media, they die off. They die. So the only kind of colonies or bacteria that end up growing on our media are going to be the ones with the plasmid. So this is why ampicillin resistance is a selectable marker, because we select the bacteria that actually have the plasmid. The next step is to distinguish between these two bacteria, right? The ones that have the plasmid with the target DNA and the ones that do have the plasmid without the target DNA. These are the ones we want. So we do that by looking at the other selectable marker, which is the X-gal, right? We said that the ones that have the plasmid without the DNA, their beta-galactosidase gene, their LAC-Z gene, is intact. And so they're able to actually secrete beta-galactosidase, and they're able to cleave X-gal and form a blue color. I don't have the color blue, so I'm just going to color it in with purple, okay? Even though it kind of looks blue on the camera. So these are going to be blue colonies. The other bacteria, this beta-galactosidase gene is interrupted by the target gene, and therefore beta-galactosidase cannot work, right? And so because it cannot work, the X-gal colorless substrate is not it's not cleaved, and it remains colorless. So these bacteria literally remain white. And so we can distinguish, like you're going to have all of these little colonies that all arise from, their one progenitor bacterial cell, okay, that are either white or blue. And we know to specifically pick the white ones because the white ones have our cloning vector with our target DNA. So we pick them, we put them on a nice fresh media so that they can continue to grow. And as they grow, as the bacteria replicates, the bacteria is not just going to replicate its genome, but it's going to replicate its plasmid DNA as well. So the progenitor not the progenitor, the progeny of the bacteria will look like this. They'll have the bacterial chromosome, and they'll also have the plasmids with the target DNA. And here you can see we're simply cloning this target DNA over and over and over again, a million billion times, because bacteria are disgusting and they grow so quickly. That is it. That's pretty much how cloning vectors work. You can see in this picture right here, the white colonies and the blue colonies. And so we'd want to specifically only pick the white colonies, right? So that's how it works. I think it's pretty genius how they even thought of all of this up. It's incredible. Make sure you go over the actual pictures, because I'm sure the pictures on the slides are way better than the pictures that I drew, even though I'm quite proud of my color coordination. Okay, so plasmids are just one of the kinds of cloning vectors that we have, and they do have many different kinds of applications. They are great because they're so easy to manipulate and use. However, they do have their limitations. One of their biggest limitations is the fragment size of the DNA. You can't really put a very big fragment of DNA inside the plasmid. Usually they can accept inserts. I don't know what I just said. Okay. They can accept inserts of up to 25,000 bases. That's their maximum. So we needed to get cloning vectors that could accept bigger fragments. They could accept larger pieces of DNA, because a lot of times, especially when we talk about human genomes and human genetics and our genes, we have really big chunks of DNA, right? We have really big fragments. So other types of cloning vectors include phage vectors. These were the ones that were discovered or used shortly after the bacterial plasmid vectors. They too cannot really hold incredibly large fragments, but they can hold fragments a little bit bigger than plasmid vectors. And essentially what they take advantage of are viruses, right? They take advantage of viruses. They put the target DNA within a virus. Usually the virus is known as the lambda DNA virus, because the lambda virus has a double-stranded DNA genome, which is what the normal living organism genome looks like, right? We have double-stranded DNA. So this bacterial lambdaphage, lambdaphage, same things. This lambdaphage we can insert the target gene in it, and then we can allow it to go through normal transduction. We can allow it to bind to and absorb to the actual bacteria, infect the bacteria with the viral genome that has the target DNA. The bacteria is going to replicate the viral genome with the target DNA and make more viruses that have the viral genome and the DNA target. So it's quite easy to use. They can carry up to 45,000 bases of DNA fragments. So still not that big. We also then have cosmids, which funnily enough are not in this. I don't know if they're even brought up, like I don't know if they're talked about in your textbook, but I'll talk about them anyway. So cosmids then are a blend between plasmids and phages put together. So they work as a normal plasmid, but then they have a specific nucleotide sequence called the COS site, which encodes all the stuff that you need to put the genome inside a little capsule, inside the little covering that normal viruses have. And so cosmids are artificially made. They're not normal. They're not natural. Like phages are natural, plasmids are natural, but cosmids are something that we've artificially made. And they can hold slightly more, like bigger DNA fragments than both phages and bacterial plasmids. Still though, sometimes we need even more bigger DNA fragments. Like for example, for the Human Genome Project, which I'm sure you guys have heard of, that was what we started doing. It was a project that started in 1990, I believe. It was a project to sequence the entire genome of the human, period. That's the sentence. Then the sentence, to sequence the entire genome of a human, right? And it was projected to last about 20, 25 years, and they finished within like 10 or 12 years. It was incredible, right? Just because DNA technology was just rapidly evolving. Now, this is what they used. They took the genome of a human, they completely fragmented, they cleaved it everywhere, so we had all these small little DNA fragments. And then each of these DNA fragments were put into an appropriate cloning vector that could hold the size of these fragments. Because remember, our genome is 3 billion nucleotides long. It's humongous. And so actually, the cloning vector that they used was a bacterial artificial chromosome, or a BAC. right? So artificial chromosomes can either be bacterial artificial chromosomes or yeast artificial chromosomes. And these artificial chromosomes act just like normal chromosomes. They replicate once, all right? And they can hold really big fragments. So they're vectors used to clone large fragments of DNA. Backs are very large. They can hold from a DNA fragment from 100,000 to 300,000 things long. However, they have low copy number. They don't replicate quite as much as the plasmid. So because they act like chromosomes, right? They don't replicate a lot in one cell. Yeast artificial chromosomes, these are really great because they're more eukaryotic. So they have their own telomeres. Origin of replication, which is actually not called ORI, it's called the autonomous replication site. But anyways, and centromere. So much like our own chromosomes, they can hold even larger fragments, but we didn't use yeast because yeasts are, since they're so similar to our own eukaryotic chromosomes, they had a problem with recombination, right? Recombination with our own target DNA, and it messed up a lot of different things. So we didn't really use, yeast weren't at least used for the Human Genome Project. We used BACs instead. But anyway, so these artificial chromosomes work the same way as the other coding vectors. You can just put them, like break them open with restriction enzymes and put in the appropriate fragment, except that these fragments now are humongous. So that was just, those were just cloning vectors. Another kind of vector is known as an expression vector. Now, these expression vectors are not, they serve a different purpose. They don't serve the purpose of cloning your target DNA. They serve the purpose of expressing your target DNA. Therefore, reading your target DNA, transcribing it and translating it, and making it into its protein. So expression vectors are really great if we want to know what the product of the gene is, right? And these are, oh, look how beautiful. I love reading plasmid maps. Oh, they're so pretty. Okay, sorry. Okay, so they're designed to ensure mRNA expression of the cloned gene to produce large quantities of the protein in the host cell. We can have plasmids, phage vectors, YACs, and BACs, and all sorts of different things can carry this gene that then gets expressed. We have expression vectors available for both prokaryotic and eukaryotic host cells. Here we have to be, it has to be a little bit tricky because if we put our target DNA, these expression vectors, like the ones that go into a bacterial host cell, are going to need to have bacterial promoters, right? So promoters with the regulatory consensus sequences that we found on bacterial, in bacteria. So like the -30 and the -10, right? Or the Pribnow box, they need those things. Otherwise, if we put the target gene from the human with the human promoter, that human promoter, even if it's found in the vector, isn't going to be read by the RNA polymerase and the rest of the protein synthesizing machine. It's just not going to be read. So we, in expression vectors, they specifically have promoters that are able to be read by the host cell that the vector is going to go into, right? Which are either bacteria or yeast. And so, yeah, we just put our target gene downstream of the bacterial promoter and then put that cloning vector, or sorry, the expression vector in a bacterial host. And then that bacterial host can read the promoter and then make the target gene into the, into its appropriate protein. So we have appropriate sequences. I can't read this because there's something in the way. We have appropriate sequences to initiate both transcription and translation of the cloned gene, which is essentially what I was talking about. And it's especially important in protein structure or function research, or make commercially valuable protein products. This is how we basically made insulin, right? Insulin on demand. We knew that it was something that we needed, and so we put the target gene in an expression vector in bacteria. The bacteria would read the gene and make insulin for us, and then we isolated that product, the insulin protein, away from all the rest of the E. coli protein, and we now have a nice little tube of a lot of insulin that we can give to our people that suffer from diabetes, poor guys. Okay, so they're absolutely incredible. And there's a lot of things that you have to consider here as well that I don't think your textbook goes into, but if it is a eukaryotic gene, remember our genes are different from prokaryotic ones. We have introns, so usually we have to make, we have to actually make our DNA into mRNA, right? So that if mature mRNA, so that we would get rid of those intervening sequences, those introns, And then we actually reverse transcribe the mature mRNA back into its gene DNA composition, except that now the gene doesn't have any intervening sequences, it doesn't have any introns. And we use the enzyme reverse transcriptase to make this happen, which is an incredible enzyme. So this type of DNA is known as complementary DNA, and I believe we do talk about it in the chapter, but I just wanted to throw that out there just in case we didn't. Oh, here, complementary DNA. Okay, so that is it for section 17.1, and I'll see you guys in the next video. Sorry about the crazy technology issues that I had.